There are two types of cells in the nervous system, neurons and glia. A neuron is an electrically charged cell in our nervous system that conveys information in our body to our brain and vice versa through electrical and chemical means. Glia are all of the other cells in the nervous system. They were both discovered around the same time in the beginning of the 19th century, but in part because glia are much harder to observe, they have been considered the glue that holds the rest of the nervous system together, which is where the name glia comes from. Neuroscience has held this belief that glia are the supporting character of the nervous system show for a while, but this is a now changing belief. So what are glia in the first place? Glia has become somewhat of an umbrella term for any cell in the nervous system that isn't a neuron. They can have many functions, from improving conductivity and protecting neurons to cleaning up debris and so on. There are a few types of glia, one of which we will discuss later, but first, how did we discover glia in the first place? And how did the notion of glia being the second character come to be? And how is this notion challenged today? Neurons have a directly measurable effect on behavior, and as such were much easier and much more interesting to study. In a squid, for example, you can poke and prod its nervous system and directly see the effects it has on its own movement. But glia is not like that, which might be the reason why it has been overlooked for quite some time in comparison to its cool older brother, the neuron. The first description of glia and neurons as separate cells is most likely French researcher Dutrochet's description in 1824. His results were a very primitive diagram that were later identified to be neurons and glia. In 1865, Dieters would describe them as connective tissue cells in his own research. 5 years later, in 1870, one of the forefathers of modern neuron theory, Camillo Golgi, would be the first to visualize neurons, glia, and their processes in the entirety. Like Dieters before him, the way they knew to differentiate the two cells is because of the lack of axons in glial cells. 5 years later, in 1875, Golgi would also give glia its first role in the nervous system, besides simply being connective tissue. The role being to carry nutrients from blood vessels to neurons, which we now also know constitutes part of the blood-brain barrier, a membrane separating blood vessels from the brain as blood is toxic to the brain. Later on, different researchers would come to produce some basic images of glia, but because of a lack of staining techniques, some very weak microscopy, and perhaps some misguided scientific judgment, as well as the fact that glia are quite a lot less exciting than neurons, the concept of glue for glia took off in the 1850s. It is not until the 1950s, 100 years later, that the electrophysiological study of glia started. Not only that, but in comparison to neurons, glia were about 100 years late in terms of biochemical studies, as opposed to simple morphological observations. By that point, the concept of glia as sort of brain glue or web that held the brain and the rest of the nervous system together was already well established. While it might seem like that isn't that big of a deal, some researchers suggest that our vision of the brain would have been completely different if glia had been studied first. For example, if our staining techniques were a little bit different. A world where university students like myself would learn about the structure of astrocytes, instead of the resting potential of neurons. As one researcher puts it, neurons, unlike glial cells, were born privileged with the result that their physiology was destined to be studied first and considered essential for brain functions and behavior. Since then, we have learned a lot more about glia. Notably, we have discovered different types of glia. I will talk about perhaps the most researched one today being astrocytes. Astrocytes, named after their star-like shape, are the most abundant type of macroglia in the central nervous system. Here are some of the features of astrocytes. First and foremost, they are a structural cell. In accordance with the glue idea from before, it is true that astrocytes play a role in structuring the brain. Notably, astrocytes support the blood-brain barrier, though to what degree they are involved, it is still being discussed, as well as provide internal structure support around the synapse. The reason for both of these being to maintain homeostasis in the brain. Homeostasis, or how I like to call it the default mode, is just the state of being where everything is nice and balanced and working well together. This includes the level of sugar in your blood, how thirsty you are, and so on. So astrocytes play a definitive role in keeping that aspect of your brain in check. They provide nutrients to neurons when they need it, call for larger quantities of oxygen and glucose when a brain region is being particularly active, and may even have a sort of nutritional reserve for themselves. Beyond that, they also work in synapses to clean up any leftover neurotransmitters and ions. You may, for example, have heard of SSRIs before, otherwise known as selective serotonin reuptake inhibitors, which are common antidepressant medications. The astrocyte plays a role in exactly that, except it doesn't seem to do so for serotonin, but instead other neurotransmitters and some other specific ions, cleaning up the synaptic cleft of any late-to-the-party neurotransmitters, and thus enabling the synapse for faster and more accurate transmission. These are all still fairly supportive roles that the astrocytes perform, but there are also more proactive roles. Astrocytes, for example, can release ATP in certain brain regions that gets hydrolyzed into adenosine, inhibiting synaptic transmission. Reasons for this inhibition may be multiple, but this process has been speculated to have an effect on long-term potentiation, the process by which our brain learns and changes with experience. At first, my reaction when I read this research was, why would an inhibitor help with long-term potentiation? That seems counterintuitive. And it turns out the reason for this is because inhibiting some synapses would enable others to make connections instead, especially some that are located in different regions than usual. In other words, astrocytes release ATP that hydrolyzes into adenosine to help the brain restructure and potentiate in different ways by inhibiting some synapses and leaving others. Astrocytes are also thought to release some other transmitters involved in long-term potentiation, including glutamate, calcium ions, as well as D-serine. Calcium 2+ ions have been thought to play a role in glial and neuronal communication as well as a bunch of other functions, but in glial aspects they seem to be a very important ion when it comes to communication. We also know astrocytes to be crucial to brain damage recovery, essentially being the scarring factor of the brain, as well as enabling regeneration when it can happen. It is also speculated that astrocytes play a role in maintaining our circadian rhythm through something called a transcription translation negative feedback loop, otherwise known as TTFL. I honestly had a hard time understanding this one, but as far as I understand, TTFL controls ours and of other animals' circadian rhythm by having something called a clock gene transcribed and translated to a protein. Now, this is genetics, but once enough of these proteins have accumulated in the cytoplasm, outside of the cell nucleus, they move towards the nucleus back and inhibit the new transcription of clock genes, creating this negative feedback loop. The proteins eventually break down and the cycle starts over again, all of this in the span of 24 hours, giving us our circadian rhythm. In mice, it was found that even in the absence of other cells, astrocytes alone could regulate a circadian rhythm through this TTFL. As you can see, astrocytes play many roles in our brain besides being structural aids. This shows that despite the history of glia preceding today's research, we are learning a lot more about glia today and proving ourselves wrong retroactively. Astrocytes are perhaps the most well-researched glia currently because they seem to dip their toes into many different parts of brain research. But there is no doubt in my mind that other glia that I didn't talk about today, like radial glia, satellite cells, and even enteric glia, the cells that line the inside of our gastrointestinal system, play a crucial role in the healthy functioning of our nervous system and body in general. If you would like to learn more about the topics I talked about today, all of the sources will be listed in the description. Thank you for watching. As you can see, there are many different functions to astrocytes, and I didn't mention all of them. This is just the research that I found by myself, and to be quite honest, there is a lot more on glia currently. But it still bothers me that neurons have held up the spotlight for so long, while glia have been such an important part of the brain, and we've kind of left them through the rug, especially in some undergraduate class where we essentially barely talk about them, even though they seem to play part in a lot of crucial functionings in the brain. As such, I just wanted to show a little bit more about what glia meant and what glia could do and why glia are cool, even though they are put up against a very other cool cell being the neuron.